This is a production of Cornell University.
Thank you, Lee, and thank you, and Dr. Nielsen for giving me this opportunity to introduce my work. So the topic today, as Lee just mentioned, I put a question mark on the, especially the genome, because I don't really know the answer. Hopefully, people in the audience give some idea. So because many of you are not working cucumber, so I will first give a quick brief introduction about the cucumber and the cucumber diseases and the molecular mapping of resistances. And then I will give 3 examples which were primarily from my lab. First, talk about the staggering gene, which mutation in this gene confers the multiple disease resistance, MDR. And then I will talk about the focus on the donor-mutual in 2 PI lines about the genetic basis, the genetic architecture and the complexity, and very diverse mechanisms. And the third example is called, we recently working on a mutation called Speckled, which it's probably not directly related with disease resistance, but it does show association with multiple disease resistance, also with some biotic stress tolerance. So cucumber, many of you may not be familiar. It's Cucumis sativus. It's also the Cucurbitaceae family with relatively small genome size, few chromosomes, 14 chromosomes, 367. It's largely cross-pollinated because it has a monoecious sex expression, but it has literally inbreeding depression. It has a short life cycle, and it has been a moderate species for a long time for sex experience and also vascular biology. There are several marketed classes as shown in these pictures. Each marketed class, marketed groups, it has a narrow genetic basis. It has been a minor crop with really very small public research community, like many other horticulture crops. But I think it is a profitable crop. The breeding work has been primarily done in the private companies. As many other horticulture crops, we have already a little bit work done even before 10 years ago, but since then, we have seen significant progress made, particularly in recent years. Talking about the diseases, I think disease resistance has always been a top priority in many crops. For cucumber, and I think many other cucurbits, the major diseases include at least major diseases here, like donor-mutual, food rot, powder mildew, angular leaf spot, target leaf spot, anthracnose, Fascimen wilt, scab garment stem blight, and also several virus pathogens. The priority really depends on the production system, which, like in the US, we usually practice open. Once we machine harvest, the donor-mutual and the food rot are actually the top priority for breeding in open field pickle cucumber production. And for some others, like the CGMMV virus, it's a new threat for cucumber production in greenhouse industry. But for some other, like a southern rotten knot nematode, it has been a problem for many years. But unfortunately, we don't have any resistant source in cultivated cucumber. So, and there have been, as I mentioned, a lot of work has been done in recent years. So molecular mapping, cloning of disease-resistant genes. This is a summary in a recent review we published, basically indicating that genes and the QTOs mapped along 7 chromosomes, cucumber chromosomes. Those red ones are the intervals of the donor-mutual QTO on different locations from different studies you can see, and the green ones are powder-mutual. you can see the majority of the studies focus on QTL mapping of donor media and powder media. There are some interesting things you can see. First, you can see the linkage of donor media or powder media resistance from their localization on the chromosomes, which is true because cucumber breeders have seen this for a long time. When you are developing a variety with donor media resistance, it also has powder media resistances. Also, for the co-localization of the facsimile view, the scap resistance and the virus resistances. Also, you can see the most many genes for these resistance are located on chromosome 5 and also chromosome 6. These are some interesting, probably I don't know that it may relate to the evolution history of the chromosomes. And of course, also there are more than 100 genes QTOs identified. Very few had been cloned. Here is a list of 15 genes which we know the candidate gene. Some of those are not really functional validated, but I think reasonably the candidate gene probably correct. Of these, half of them are donor media and powder media resistant. You can see the donor media, this column are some of the functions, 3 of the staggering gene, which contribute to resistance across 3 pathogens. And also donor media, you can see all these 3 for donor media resistant clonate, all of them are different. There are 4 genes for powder media, which are people are probably more familiar with these kinase or MO genes. So I'm going to focus on the staggering gene and focus on donor media and also for the multiple disease resistance, as I mentioned, for the speculative mutation. So for donor media, many of you probably familiar, particular people on cucurbits, which is the biggest threat of all the cucurbits, but it's particularly a problem for cucumber. It's on the side, the pathogen called Pseudospora cucumbens. The primary reason for this develops so fast. Once you find it, it's very difficult to control. You can see the symptoms of donor media. I want to emphasize that there are the chlorosis, necrotic spots. Based on the symptoms, we also, when we are doing phenotyping, we can use different criteria like sporulation, like here, or necrosis. Sometimes we call it collapsing, chlorosis. We can individually score for the symptom development using 3 criteria, but in most cases, we just use called a general impression, which is a summation of the individual symptoms. So these are very important to understand the underlying genetics of these resistance for donor media. So donor media has an interesting breeding history, which was first reported in 1868 in Cuba, actually. That's why it's called Cubensis. But the breeding work started here in the US in late 1940s. Actually, the first cucumber variety called Pamitu was released in 1948. Unfortunately, it was brought down 2 years later. Since then, the cucumber breeders spent a lot of time developing donor media resistance. And in the 50 years in 2004, we have been focused on use a single source of donor media resistance from the PI 19737. We call it, it's a single gene called Yiqun Weng, like the pond set series, which was developed by Cornell cucumber breeder. It's very famous, which carries a Yiqun Weng. Also, we have very well-known germplasm called GY14, and WA2757. These two genes are very popular for cucumber breeders, which carry the DM1. Since 2004, the DM1 was no longer effective. Fortunately, we identified a new source of resistance for the new strains of particular to PI lines, PI 197088 and PI 330628, with high resistance. So these 4 lines are the major targets of my presentation today. First, I'm going to talk about the downimage resistance in the old downimage, we call it 2757 and the GY14. The 2757, it's really a legend, which has resistance to 9 passages, which was developed by my predecessor, Dr. Clint Patterson. After the breakdown in 2004, North Carolina State screened the collection, and in the cucumber varieties, 2757 still maintains a pretty good resistance among the varieties. So we have interest to do QT mapping in GY14 and 2757. We used the classical QT mapping studies. First, we used the real population from GY14 using the Elwing called Colossus and the GI. We scored at the different time points in multiple environments. You can see in all the experiments, we detect a single QTO, which was located on chromosome 5, which is consistent with the previous study. So that's the DM1 we have been knowing for a while. Interestingly, when you use the Elwing, it's in this population, at different time points, this QTO was consistent detect, but if you use general impression, sometimes when you later, the first time you have a high, you can detect the QTO, but at a later stage, it's going to disappear. So that will says two things. First, the criteria we used is important. Second, the time of scoring also very important. But anyway, in this case, GY14 contains a single QTO, which is DM1 or chromosome 5. But and also, it basically it's an anti-chlorosis plays the important role in the downimage resistance. But in the 2757, depending on which criteria you use, if we use Elwing, you only detect DM1. But using necrosis, basically, you only detect a second QTO of chromosome 5, which we name it DM5.2. But if you use GI, which is a summation of chlorosis necrosis, you're going to detect both, of course, which depend on what time point you're going to you score. So in 2757, there is additional QTO in addition to DM1. This is the conclusion from QTO mapping studies. But in previous breeders also find that the 2715 and GY14 also have resistance to the anthracnose. So at that time when we started work, we have no idea they are actually the same gene. So we have an independent study to map the anthracnose resistance in the 2 lines and use a QTO mapping strategy. True, it's also mapped in chromosome 5. We name it the CRA, but in 2757, there are additional minor QTO. The same is true for angular spot. And we've identified the angular spot resistance in GY14 on chromosome 5 with the same location as the DM1. But in 2757, we found additional QTOs. So if you do joint QTO analysis, you're going to find the DM1 PSO for angular spot and the anthracnose are all at the same location. gene. So we continued the fine mapping and map-based cloning. Eventually, we will be able to identify the candidate gene, which is a staggering gene. Staggering, it's called mechanism dechalates, plays important role in the chlorophyll degradation. So we examined the symptom development, the pathogen growth, and the staggering gene expression. You can see on the susceptible lines, it has a typical chlorosis symptom. The pathogen keep growing, population keep increase, but also in the susceptible line, this gene shows significant increase of expression, but in the resistant lines, it has a low level expression. So consistent with our expectation of the chlorophyll degradation pathway may be involved. Also, if we do the phylogenetic analysis, we can see this gene which was originated from PI 197087 from India is true. This staggering gene, the mutant allele was originated from PI 7087. Yeah, if you do using polymorphism in a Kinetic KB region to do phylogenetic analysis, you can see all the resistance will be clustered in one small clade. Why we use Kinetic KB? Because the AOD in this region is very high, very low, with high recombination. This is really consistent with the breeding history because all the resistant lines carrying the DM1 in the US breeding materials carry the haplotype basically, and those susceptible ones don't. So that means this gene was under selection during the breeding of the US pickling and slicing cucumbers. And we examined all the genes in the chlorophyll degradation pathway among 3 lines, including the 2 resistant line and a susceptible line. You can see consistent with the staggering, other 2 genes downstream of the staggering gene, PPH and the PO, showed similar expression patterns, but those on the chlorophyll AB biopath are a little bit different extreme pattern. These expression profiles suggest that true that these resistance indeed is involved with chlorophyll degradation, but how we don't have, we are still working on this. And here is basically our speculation that in the susceptible vein, we have regular chlorophyll degradation upon infection of the disease pathogen, but and you have typical symptoms, chlorosis, necrosis, and the disease. But in the resistant one, because of mutation, a single mutation, yeah, this pathway was less efficient in degradation because the symptom development staggering is required. If this mutation will block the chlorophyll degradation, which you show the resistance. Actually, it's a passive resistance, but we don't know if there is any active resistance because we can see the different reactions upon infection in the anthracnose, stony midge, and angular spot. So we speculate probably there are some active defense during the interaction. But a lot of more work we need to do. So that's the staggering story. Next time we talk about the Q2 mapping studies in 2 PI lines. Upon that, when you large-scale screening using the 2 new strain, the 2 PI lines, PI 197088 and 30628, should high resistance, but commercially, they're commercially not acceptable because it's very wide locking. 33068, also called 7120WI, only has donor immune resistance, but the 7088 has both donor immune and polymerase immune resistance. So we conducted a QTO mapping studies in the two lines and identified two major QTOs in 3300628. And we identified 11 QTOs in 7088. And also we identified the 4 QTO for polymerase resistance in 7088. It's interesting to see, based on their chromosome locations, all the 4 polymerase QTOs are co-localized between the two PI lines, suggesting they may be the same. But of the 11 QTO in 7088, 3 actually contribute to susceptibility. And there are 2 unique major infect QTO in 7088. So you can see there are fewer QTOs in the 3300628. That means from breeding perspective, probably this PI line 33006 is better than the 7088. And also for donor immune and polymerase in the 7088, you can see the 4 QTOs also co-localized. That also suggests the linkage, basically, this line for the both donor immune and polymerase resistance, because for DM4.1 and the DM5.2 are co-localized, which are major, in fact, QTOs in both lines. So we focus the 2 QTO form fine mapping. The strategy is to just first we developed NEOs for individual QTO to mandelize basically the individual QTO, and then we do segregation linkage analysis in multiple year, multiple location, phenotyping, and that's, I think, most of us are familiar with standardized. So we are able to eventually narrow down to a very small region using multiple year phenotypic data. In the case of 4.1, 82 KB. That's based on the 2018-2019 data. But in 2020 data, there are a couple recombinants which are not consistent. The candidate gene region shift to a different location. So that's always true when you're working with QTO with relatively not so large in fact. That we learned some important lessons. But actually, that's not necessarily something wrong, because recently another group working in PI 7070-197088 on the DM1, they suggest there are actually some subacute, 3 subacute at the DM4.1 locus, and which are very closely close with each other, and they identified the candidate genes for the 4.12, which is a receptor-like kinase, and the 4.13, which is an amino acid transporter, and the polymorphism is both the 551 BP insertion or transferrin insertion. So we examined our data in 7120, as I mentioned before, the 2 lines at the 4.0 locus, they probably share the same. It is true, but we further analysis, we think probably there are additional QTO, sub-QTO, we call the 4.12A and the 4.12B. We examined the allelic diversity of the 2 clonal QTO 4.12 and 4.13, and that this 7120 line also carry the variant. So we think they are the same. We are focusing on clone the 4.12B and 4.11. This is probably reasonable because 7120 was from Pakistan, originally 71088 from India, which geographically not very far away, could be reasonably speculated they have the same origin. And for 5.2, we use the same strategy. And at this time, we narrowed down 50 KB. I don't think we have anything wrong here. And we have a couple of genes we think it's very possible, but we need more data. It's likely a phosphate transporter. And another interesting thing in 7088, the same QTO also, the polymerase 5.2 resistance also same location, likely the same gene contribute to both dominant and polymerase resistance. That's our speculation. The third example is about the Speckled, which probably associated with the multiple disease resistance. We identified this is visually, we call it the speckled leaf because there are many chlorotic spots at the first, second, third true leaves. But after the fourth, probably usually in this particular line, after fifth, when the plant reaches fifth true leaf, those chlorotic spots will be gone. And we conduct genetic studies, and this mutation is controlled by a single recessive gene we call SL for Speculative. And we grew these lines in field. That's really intention we never found. But last year in our field trial, which we have very serious angular spot in the field, we find difference between the mutant, its original donor line, we call that W means wild, means mutant. And we calculated the mean disease scores. It's a significant higher disease scores for the wild type. And we find that at the end of the season, we measured the fruit-related traits. We find there are no different, a significant difference. Basically, the mutant perform much better than the wild type under the disease procedure. At the same field, it grew different populations. We found all the plants carrying the mutant line have better resistance than those plants carrying the wild alleles, which is true in different populations. This suggests that the resistance is truly is kind of independent of genetic background. So based on this, we did some artificial inoculation tests in the greenhouse growth chambers. For example, we tested the anthracnose, and you can see after even 1 week of inoculation, the mutant still looks pretty nice and healthy, but the wild type almost dead. So we did follow tests within polymerase, dominant and the target spot, which showed similar trend. That's for the disease resistance, but for the phenotype, we found some genetic background effect, like if we introduce that mutant allele into another cucumber line called 9930, and we find the symptoms of the mutant phenotype is much more strong. Usually in the original line, the symptom, the chlorotic spots will be gone when it reaches fifth true leaf. But in this new background, even 10, 15, 20 true leaves, you can still see even the flowers and then the fruit skin. You can see those whitish stripes. So that means this gene probably is expression affected by the background. And accidentally, we also find that this mutant had better tolerance to water stress because the grass student forgot the water when they put the plants in the greenhouses, and maybe a few weeks a few days later found that actually the mutant seedlings still feel happy even under very severe water stress. So we conducted experiment just progressively with water withdrawal and locked the appearance of the mutant and the wild types and measures the relative water content. You can see at two different temperature settings. The mutant, yes, it keeps higher water content and not less wilting compared with wild type. We also did salinity stress experiment using different concentrations of sodium chloride, and we have similar results. So basically, what we see is the mutant line has a better resistance to both disease resistance, multiple disease resistance, and the about stress tolerance. So we clone this gene and find a retrotranscription insertion in the first exon of the candidate gene, which results in the deletion of the 72 BP transcript in the cDNA. And we did subcellular localization of the protein, find it's located in the chloroplast. The RNA-seq transcript profiling in the mutant and wild types, we find that the top 4 GUR enriched terms are all related with the membrane biogenesis. So this may suggest that this gene may function in biogenesis because it's located in chloroplast, some chloroplast biogenesis. More specifically, in protein transport from nucleus to the chloroplast. So there are multiple pathways we know of. Particular is CPSRP 4350 pathway. So there's a protein synthesized in the nucleus, which will be transported into chloroplast, first passing to the double layer of the chloroplast from the TikTok machinery. Then, after entering the stroma of chloroplast, the SRP pathway will deliver the protein into the syncyloid of the chloroplast through the SRP54, so the 2, and then the inserters and the receptor and inserters. So we examined the protein-protein interactions with Y2H of all the components in the pathway with the speculative protein, and we find the interactions. But if we there's some reduced interaction when the mutant allele is used for the interaction study. Many of those interactions were validated with BIFC. So how this mutation is related to the multiple disease resistance and the drought tolerance? So I don't have any evidence. We are still working on it, but that's our speculation. Probably because the gene functioned in the delivery, particularly for the HCP proteins, which were important for the photosynthesis and the biogenesis of the syncyloid membrane. So if the mutation may affect the transportation of those HCP proteins, which may disturb the photosynthesis. So this is a graph from a review publication indicating the pathways in the photosynthesis system 1 and system 2, where the reactive oxygen species are produced in different steps. So we speculate this disturbed photosynthesis system may increase the production of IS. So indeed, we we measured the ROS in the mutant and white lines. That's correlated by standing of the oxygen reticle. You can see the much higher level of the ROS in the mutant line. So here is our hypothesis. Basically, it's well-known, well-established ROS involved in almost all those biotic biotic stress tolerance resistances. So this mutation may be related with the ROS production, which may also increase the ROS basically signaling for both abiotic stress tolerance and biotic stress. These resistances. We probably need more evidence. That's our hypothesis. So that's all for I have for the 3 examples. And first, lastly, some of my thoughts or speculations. So if we have the 15 cucumber R genes or candidate genes cloned, you can see most of them are recessive. And you can see also the strong linkage, particularly for donor-MITU and polymerase resistances. And you can see there are very few MBR type genes. So several years ago, there is a review of the R genes in plants. There's, as of 2018, there's 314 R genes cloned from plants, which based on the functions and the mode of interactions, they could be classified into 9 classes. Broadly speaking, based on the proposed by the authors, they're perception-based. All those kinases like AR, NBR type, receptor kinases, receptor proteins. And also the second category is loss of susceptibility genes and some others. So based on these classifications, the 15 genes we can put under or 3 for the perception-based, like the YK war-assisted kinase, which we cloned recently, but we didn't publish that yet. And the receptor-like kinase or MBR type, there are 4 genes, belong to 5 genes. For the loss of susceptibility, we have the staggering gene, as I just mentioned, the MO for the polymerase resistance, and also the amino acid transporter gene for the DM4.13. For others, like the virus resistances, for the, I don't know, the Speckled belong to which category, but they look at the percentages compared with the other plants. In all the genes cloned so far, 80% belong to the perception-based MBR receptor-like kinases, receptor-like proteins. 15% belong to the loss of susceptibility. So if we compare cucumber with those in other plants, you can see the distribution very different. So that's why I say, okay, there's some special things, but why? I don't need the answers, but here are a few of my speculations, which many of you probably have a better idea. Of course, compared with many other plants, one reason maybe we only have 15 genes cloned, compared with Arabidopsis 157 or more, rice 120, tomato 70, potato 45. The sampling size is too small, which is probably true, probably not. Of course, it's true. There are few MBR genes in the genome, which is true for all the cucurbits, because you have very few MBR. Those genes are large reservoir of new genes for generation of new genes. We don't have many numbers. So the plants probably have to explore other mechanisms. And also, I guess the single domestication and the domestication bottleneck and very limited secondary gene pool, sex compatible relatives, and the genetic basis within each group or the breeding practice also have something to do, because it's all a private business largely. They always focus on a very narrow genetic base or gene pool. We don't lack resistant sources in our collection, but the breeding practice really use those, and unless you have really urgent need to explore because of the breakdown or no emergent appearance of new diseases or new races, strains. So that probably has something to do. Talking about the NDR, here is the phylogenetic tree of the cucurbitacea. You can see through all the cucurbits species have relatively few NDR genes. In most cases, 70, 80, but in wild species like hystrix, a little bit more. So the spongy gold is the only anomaly, which I'm not sure it's correct or not. But you compare with Arabidopsis, 250, tomato 265, rice even more, 535. So that's probably one reason. Another thing which I have to say, yes, the primary gene pool is a wild cucumber, Cucumis sativa hadwicae. Wild cucumber actually, you have a free gene flow with cultivated cucumber. We don't have any real valuable source of resistance from wild cucumber, which is distributed primarily in India, Thailand, and the surrounding regions. Cucumis hystrix is the only species in Cucumis which have 54 species, which is actually compatible. But in nature, you cannot cross. So that's really limit the use of the relatives as a source of resistance. And the cucumber is the only species in Cucumis with only 14 chromosomes. All the rest have 24 chromosomes, 48 chromosomes. So evolutionarily speaking, which is very different from like tomato or potato, which have a lot of relevant relative species. So in this sense, the 14 chromosomes was involved from 24 ancestors through descending diploidy. In my opinion, it's evolutionarily speaking, it's a dead end for evolution for cucumber. So that probably has something to do. So that's all I have for my talk. Lastly, I would appreciate the work report today. Many people contributed, including students, graduate students, postdocs in my lab, and also collaborators from both private and public institutions. My work is supported by both USDA and also from industry. Thank you for your attention.
Thank you, Yiqun, for the presentation. Are there any questions from the audience? How about I start with the first question? Yiqun, it's really interesting to find you cloned the state green gene as a DM resistant gene. The state green normally it's involved with delayed senescence. So do you know in any other crop state green genes also involved with some aspect of disease resistance?
Yes, at the beginning, because that's the first time. Some time ago, I think in Arabidopsis, staggering was shown to be required for symptom development. And then we find that it contributed to multiple disease resistance. We are really not sure, but yes, in soybean, Michigan State, they proved that they published a couple of years later, I think it's last year, 2 years ago, in plant physiology, they showed that in soybean, the staggering also contribute to disease in soybean.
I see. So a comment from Mike: serendipitous discoveries are always the best. Right, fully agree with you, Mike. And from Rebecca, thank you for the wonderful talk. And again from Mike, the mechanistic story involved chlorophyll is impressive. Enjoyed the talk. I wonder, are there any additional questions?
I do have a comment. Thank you for the comments. In both staggering and in the Speckled cases, I think the chlorophyll, the photosynthesis pathway plays an important role. We did some preliminary studies, and we are accumulating evidence to suggest the photosynthesis components, particularly the HCP proteins, probably involved in both cases for the resistance, because we see the interaction between the chlorophyll, the HCP proteins with both the staggerin and the speculative proteins.
Yeah, your speculative story is really amazing. So I guess for all the speculative, if they are involved with some resistant lines, they should show the speculative genotype, right? Phenotype, right?
The speckle phenotype will be gone when the plant passes fifth leaf stage.
Yeah.
So
I'm just curious, if you look at the germplasm of cucumber, say if that gene is also associated with general resistance in cucumber or not.
we didn't find it. There are different mutants on this. Some mutants show the yellowing or speckle, but not like in Arabidopsis. I think there are a lot of mutants called variegation, which is very similar to what we see in cucumber. Theoretically, we can also call this a variegation mutant. But first, they're controlled by different genes. I haven't seen the same gene, same phenotype in any other plants for this, for the same.
Right. So there is a question from Mike. Mike, do you want to ask directly or you want me to read it?
I can read it. So is the photosynthesis component important for adaptation first? Then it evolved to have a disease resistance component second?
Thank you for the question. I'm not really, I don't think this mutation from plant, not from breeder perspective, from plant perspective, they have any intention to be associated with diseases. So from evolutionary perspective, yes, they're probably not. But that's the elevated level of OS is a consequence of the disrupted photosynthesis system. I'm not sure it has any adaptive consequences for plants.
Okay, great. Thank you for your answer.
Jeff, let's go with Shatarias first. Shatar, do you want to ask your question directly?
Sure. Yeah, thank you for your talk. It's a very nice one. And stay grain trait is associated with drought tolerance in some crops. So does this mean that the same gene is involved with downy mildew resistance? And what the mechanism? If
Oh,
that is...
sorry, staggering is not associated with drought tolerance. The speculative mutation is. Staggering is not.
Okay, thank you.
Jeff?
Sure, yeah, thanks for the talk. I just was intrigued by the low number of R genes, and you may have said more about this, but is there anything about the biology of that family that might explain why the low R gene number is true? And I was wondering if it's anything to do with the autoimmune response that's usually given or is sometimes given as a reason for why larger numbers of R genes are not beneficial.
First, the low number of by R gene, we are talking about, in most cases, talk about NBR.
Yeah, right. Mm-hmm.
It's true, NBR genes relatively fewer in the R cucurbitaceae family. The reason people speculate is because the whole family lacks of the recent whole genome duplication. So that which is true. The whole cucurbita family relatively has, except for the cucurbita, the pumpkin squash. But overall, even in pumpkin squash, we're even half of the number of NBRs compared with the cucumber or melon. So overall, this family has few compared with others because lack of the whole genome duplication. That said, as I mentioned, it's potentially that this family could explore other mechanisms like the receptor-like kinases, receptor-like proteins. We don't know because so far we have very few genes, R genes cloned in the whole family. But some people speculate maybe there are more genes for RAK or LP genes. And of course, for the simple 15 genes, there are unproportional high numbers of other categories or the loss of susceptible categories. My guess is yes, because they don't have a lot of R genes, they have to explore other ways.
Right. Yeah, no, thank you. It's an interesting question.
Yeah, thanks.
Okay, so you consider the time the last question from Rebecca. Rebecca, you want to ask directly?
Sorry, I'm muted, double muted again. I'm sorry. So a couple of questions, really, if I can squeeze them in. Why do the plants outgrow the speckled phenotype? And also curious, and why were the leaves actually smaller as they appeared to be in one of the figures for that phenotype? And then thinking of the stay green, is it really fixing a general defect with the rest of the germplasm? You know, this excessive buildup of reactive oxygen, is that sort of a general problem with the germplasm that you fixed with that stay green?
It's an excellent question. First, for the speculative mutant, it does have some I call side effects or negative linkage drag or whatever. Yes, it's relatively slow, particularly at the beginning of an earlier stage of seedling stage, slower, small leaves. But at the later stage, it gains the growth compared with the wild type, particularly under disease pressure. It definitely it grows much more healthier, faster, with as I showing that it's performed much better. So why it's gone that after like fifth leaf? In plants, there are one class of mutants called worsened leaf or worsened mutant. It's very common in many plants, like the yellow leaf. All the new leaves should grow yellow, but the color will turn green after when they become mature, or they show yellow cotyledons or yellow. They're gone. The same is true for the for the speculative chlor spots. This probably, there are some series, particularly working Arabidopsis on the fluorescent leaf mutants, and also that probably have some interaction during for the chloroplast translation control. We are actually working on it, but I don't have an immediate answer for this. But it has something to do with the translation of the chloroplast machinery. We have some evidence if we use chemicals to inhibit the translation, we'll be able to return the phenotype. It's going to be gone. And what's your second question?
Well, I was wondering if the stay green is really fixing an actual problem, like a mutation or some problem that's in the majority of the germplasm, where you have that buildup of reactive oxygen that's excessive and causing chlorosis. I mean, is it better to seen as a mutant or as a fix of a mutation?
First, this mutation, yes, we did some of the RNA-seq analysis for the staggering. Constitutively, the photosynthesis machinery, some genes involved in photosynthesis will be differently expressed between the staggering gene and the wild type. But this one will not affect the general performance. We don't really see any significant differences in performance, in growth, or between the staggering mutant and the wild type. I would say it's, in this case, it's adaptive trait, because this has been existing in the wild germplasm collection for a long time. It probably, this mutation, okay, I think it's also relevant to the IOS elevated IOS level to fight the disinfection, which is happened to be explored by the plants and found by the human being.
Okay, so let's consider time. Let's stop here, and please join me to thank Yiqun for the wonderful presentation. Thanks, Yiqun.
Thank you all.
This has been a production of Cornell University, on the web at cornell.edu.